Hi everyone, my name is Rachel Kosick and I am a second year student in the Cell and Molecular Biology program here at the University of Wisconsin-Madison. So I'm originally from Maine, but I got my undergraduate degree in molecular biology from the University of Pittsburgh. From there, I came straight out of undergrad to grad school here at UW. Within the CMB program, I'm in Audrey Gasch's lab in the Department of Genetics. So in the Gasch Lab, we're interested in understanding how eukaryotic cells sense and respond to environmental stresses. We use budding yeast, or Saccharomyces cerevisiae, primarily to explore this, but we also look at other species of yeast to try to better understand the eukaryotic stress response as a whole. To explore this, our lab employs a lot of different experimental tools, including microscopy, transcriptomics, transcription factor analysis, and comparative genomics and proteomics. Although we focus on the eukaryotic environmental stress response, there's a wide array of different projects going on in the lab, and if you join the Gasch Lab, you have a lot of input in selecting the focus of your work, and this rings true for a lot of other labs within the program. In my own project, I am studying the role that protein kinase A, or PKA, plays in the yeast stress response. I'm exploring the PKA pathway's effect on transcription and gene expression during stress. Specifically, I'm investigating the interplay among PKA, proteins that regulate PKA activity, and transcription factors at chromatin, and seeing how this regulates transcription. Right now, I'm working on a large ChIP-seq experiment to explore this. Overall, I love being in the Gasch Lab. Everyone in my lab is very supportive. We get to do pretty amazing science, and we have a lot of freedom to design our own experiments and explore what we want to research. I chose to attend graduate school at UW-Madison for a variety of reasons. I was first drawn to the program because I wanted to join a large umbrella program with tons of different research opportunities. When I visited for interview weekend, it pretty much sealed the deal that I wanted to join the CMB program. All of the faculty and students I met made me feel really welcome, and everyone seemed really content and happy living and working in Madison. Personally, I felt really comfortable on campus, and I had such a fun time during my interview weekend, learning about all of the research here and interacting with the students. Since moving to Madison last year, I've absolutely loved my time here. I've made a great group of friends within the CMB program, and they've really helped me feel at home here. Outside of the lab, I love spending time outdoors, especially hiking, and watching sports, mostly hockey. I also really like to cook, and I found a lot of incredible farmers markets around Madison. And before COVID, my friends and I also spent a lot of time exploring the local breweries. Overall, my experiences in Madison and at UW have been amazing, and choosing the Cell and Molecular Biology program to obtain my graduate degree has been a great decision.
Hi, I'm Ellen. I am a fourth year in the Cell and Molecular Biology grad program, and I work in Audrey Gasch's lab, just like Rachel. Originally, I'm from Ohio, but I did my undergrad at Ball State University in Indiana, where I got my BS in biology with concentrations in microbiology and genetics. So in the Gasch lab, I also work with studying protein kinase A and yeast, just like Rachel, but I tackle this from a different perspective. So half of our lab actually works in the Great Lakes Bioenergy Research Center, which is based on campus. The name is a mouthful. And the goal of the center is to develop methods to more efficiently produce biofuels. And so my project is working with a strain of yeast that has been engineered and evolved for biofuel production, and it has a mutation in the protein kinase A pathway, and it causes this weird phenotype where the strain can ferment the sugar xylose. Xylose is one of the fermentable sugars that's released from plant biomass. So this yeast strain can ferment xylose, but it doesn't grow, which is very weird. since usually fermentation leads to growth. So my project is investigating how this mutation in the PKA pathway has altered the physiology of the yeast to allow it to ferment without growing. To tackle this question, I am doing a variety of omics experiments. Right now I'm working on RNA-seq analysis. I'm also going to be doing a proteomics experiment looking at protein-protein interactions in the PKA pathway, which is just kind of a sample of what we do in the Gasch Lab. As Rachel said, we do a lot of omics works to characterize how a cell changes globally to respond to environmental changes. Now, outside of lab, other things I really like to do is to be outside. Madison is such a nice city. For that, we're a very active city. There's a ton of paths that you can go biking on or walking on. It's great being by 3 lakes. Over the summer, I got a kayak and went kayaking a bunch, which was so much fun just to spend the whole day out on the water. We're by a lot of really nice state parks. So if you're interested in hiking, it's great for that. But I also love to bake, and so I do bake a lot and then bring the goodies into lab so that I am not eating them. And being in Wisconsin, we have a lot of breweries to enjoy. Right now, most of them have limited capacity, outdoor seating only, but they are still open. And you can also buy their beer in stores and just enjoy it at your own house. So that's kind of a taste of what I like to do here in Madison. There's so much going on in the city. Even with the restrictions due to coronavirus, there's still a lot you can do outside, socially distanced and safely. And yeah, it's a fun city. UW-Madison has been such a great university to attend. And the C&B program is also a lot of fun. Everyone in the program is just tons of fun to hang out with. So I hope you enjoy searching for grad school. And so next we're going to take you on a tour of the Gasch Lab. We are going to take you through one of the labs in the genetics building here on campus. This way. So what's pretty nice about our lab space here is that we have separated desk area and a separated lab space area. So right now we're in kind of our office desk space, and this way, if you turn, is where our lab is. It's kind of a fishbowl where there are windows everywhere that you can watch people work as you sit at your desk. And so here is our desk area. We each have our own desk and we have our own computer. It's really nice. We can eat and drink out here. We don't have to worry about that. We have a lot of shared food, especially during lab meetings. A fancy tea station, which is great. So going into the lab here, this is an open lab space. So we share the lab with 2 other labs. So this is a separate lab. And over here is where we work. We have our gel loading space, fridges and freezers. What's really nice right now is that we all have our own bench and bay because of COVID and social distancing. Before, there were a few of us that had to share our own benches, but now we each get our own bay, which is nice. We can spread out a little more. We have our thermocyclers, fume hoods over there. We've got a couple of microscopes. We have just a plain old light microscope here for dissecting. Then we have a fancier microscope in that box. We use it for fluorescence work. That's why it's in the box, so no light gets in. We have incubators, chemical shelves. And then we have our -80 room in another incubator. And then we have our autoclave dishwasher space, followed by the cold room here. So that is the Gasch Lab here in genetics. Thank you for walking on this tour with me. Hope you enjoy this and all the other lab tours you'll get to see.